Good morning students. So today we are going to discuss the second chapter of class 12. If you haven't gone through the first chapter, I will recommend you to go to the first chapter, see the videos of the first chapter and then come to the second chapter. The second chapter is sexual reproduction in flowering plants, where we'll see the plants how they reproduce sexually by means of producing gametes, and then these gametes fuse to form seeds and corresponding fruits. I am your biology teacher, Ankesh Singh. You can reach me at these two places, whichever is feasible to you, for clearing your doubts and suggesting anything. Let us move ahead with the chapter. The first thing comes are the pre-fertilization event. Now, as we remember, the sexual reproduction can be divided into three different categories: pre-fertilization event, fertilization, and post-fertilization event. So we'll see the pre-fertilization event first in case of the plants. Several structural and hormonal changes brings about these flowers on the plant. These formation of the flowers, buds, and then the flowers inside a plant is known as inflorescence. Flowers are the reproductive part of the plant. In case of the flowers, we can see two different structures. One is the male structure that is known as androecium, and the second structure is gynoecium, which is the female part of the flower. In the diagram, you can see the different parts of the flower. Here you have stem, calyx, then the petals or the corolla. Then comes the androecium or the stamen, which contains anther and filament. And finally, the gynoecium, which contains stigma, style, and ovary. So let us first discuss about the male part, that is androecium. The androecium is the male part of the flower. As we have discussed earlier, it consists of what? Filament and anther. Filament is a long cylinder like structure, and it is the stalk which holds the whole androecium or the whole anther with the flower. Anther, whereas, is a bilobed structure and it is found at the top of the filament. The filament is attached to the thalamus or the petals. So it is either attached to the gynoecium part, or it is attached to the petal, or it is attached to the base of the flower. You can see in this diagram the red colored section are the filaments, whereas the bubble like sections which are present at the top of it is nothing but it is the anther. Let us first see about the anther, because we have discussed about filament previous also. What is filament? It is a stalk like arrangement which is attached either to the thalamus or to the petals of the flower. What is anther? A typical anther is bilobed, and each of these two lobes contains two theca, and hence they are known as dithecous. This theca is separated by a longitudinal groove which is running in the lengthwise direction. The microsporangia, the cells which are located inside the theca, they further develop to form pollen sacs, and these pollen sacs contains the pollen grains. You can see in this diagram, this section is known as the anther. It contains two lobes. Each of these lobes contains what? Microsporangia. These microsporangia further develop to form what? Pollen grains. Next we have the structure of microsporangium, from where the pollen grains are formed. The microsporangium is surrounded by four walls. You have to remember this one very well. This is one of the most hotspot area of asking questions. It contains four walls: epidermis, endothecum, middle layers, and teptum. The three outer layers, that is epidermis, endothecum, and middle layers, helps as a protective layer for the inner spores what we have. It helps in the dehiscence of the anther and release of the pollen grain, whereas the teptum helps in providing nourishment to the pollen grains. The young anther, the sporogenous tissue, in case of the young anther, when the anther is very young, the sporogenous tissue which is present in the center of each sporangium. At the center of each sporangium, what we have? We have sporogenous tissue. From this sporogenous tissue, it will form the pollen grains. Outside that, what we have? Teptum, then comes middle layer, then endothecum and epidermis. Epidermis, endothecum, and middle layer serves as a protective layer, whereas teptum helps in providing the nourishment. You can see in this diagram, epidermis, endothecum, and middle layers serves as a protective layer. Teptum, on the other hand, provides nourishment, and inside what we have? Microspore mother cell, or pollen mother cells, sometimes we say. Now let us talk about how this microsporangium develops the pollen grain. Just like in case of gametogenesis, what is it? The formation of the gamete from the diploid mother cell. Similarly, that process here is known as what? Microsporogenesis. Why? Because it is synthesized from the microspore to the pollen grains. The process of formation of microspore from the pollen mother cell is known as what? Microsporogenesis. As the development occurs inside the anther, the sporogenous tissues undergo meiosis to form a microspore tetrad. Each of the sporogenous tissue gives rise to this kind of tetrad. Whatever the sporogenous tissue cells are present inside the microsporangium, all these sporogenous tissue develops a tetrad. As the anther matures, the microspores get detached from each other and it generates individual pollen grains. You can see in this diagram, the first one is microsporocyte, which is a diploid nucleus. Then it undergoes meiosis once, and then it undergoes meiosis 2 and develops 4 nucleus inside a single cell. Then all these 4 nucleus develops 4 different cells, which is known as tetrad. Each of these are gametes. Then each single tetrad is released in the form of microspore, and it undergoes several things and it develops something known as pollen grain. These pollen grains, these mature pollen grains goes and further fertilizes the female gamete. Pollen grain. Let us see the pollen grain. It is represented as the male gamete and it has 2 layers. One is exine, that is the outer hard layer, so that it can withstand very extreme drastic weather conditions. The inner layer is known as intine, which is thin and a continuous layer, and it is made up of cellulose and pectin. The mature pollen grain contains 2 different kind of cells. One is known as the vegetative cell, containing a large irregular nucleus, and it is the food reserve. And the second one is generative cell, which is rather comparatively small and floats inside the vegetative cell. This generative cell is the cell which goes and fertilizes the female ovule. The viability of the pollen grain when they shed from the parent's body depends upon the temperature and the humidity of the surrounding. If the temperature and humidity is feasible, it can fertilize very fast. If it is not, it will take some period of time. The time till which these pollen grains become viable, that means they stay alive, can vary from 30 minutes to few months. You can see the diagram of a pollen grain. The outer layer is known as exine, the inner protective layer is known as intine. Then we have the generative cell inside that particular pollen grain, and we have a vegetative nucleus. The whole cell is known as the vegetative cell. Inside that vegetative cell we have the generative cell. So what is the summary of today's lecture? We have seen about the androecium, we have studied about anther. Furthermore, we have gone through structure of microsporangium. We have discussed about microsporogenesis, and finally we have seen pollen grains. So these sums up today's lecture. Thank you all for listening this lecture very patiently. Hope you all understood the things. If you haven't, if you have any questions regarding this matter, you can reach me at these 2 places, whichever is feasible to you. And this completes today's class. Until the next time, stay happy and stay safe.